This is Chris. He's a stage 4 lung cancer patient, and there's not a lot we can do for him, unfortunately, because a lot of therapies will kill him before they kill the cancer. So how can we save him? In order to save Chris, our cancer patient, we must first understand how immunotherapy works. We can start by imagining two cells, a normal cell and a T cell. These cells can't actually see each other, so they can only identify each other through touch. Let's zoom in on some membrane-to-membrane interaction. The normal body cell will express membrane identifier proteins that indicate how healthy and what kind of cell it is. And the T cell will have an antigen receptor which can scan the proteins to check the cell. The T cell extends the antigen receptor and it checks the normal cell and leaves it alone if everything's all good. If the cell is cancerous or unhealthy, then it will usually express faulty proteins, and when the T cell recognizes it, it will flood the cancer cell with toxins and destroy it. However, on rare occasions that the cancer cell is able to produce the correct protein and the T cell lacks the capability to identify the cell as cancerous, then nothing will happen and the cancer will proceed to divide uncontrollably. This is cancer and this is bad, but there's a solution. This is called chimeric antigen receptor T cell therapy. We begin by extracting T cells from the patient and then we place them in a lab-grown environment with genetically modified viruses. These viruses will contain the code for the chimeric antigen receptor, which can identify specific proteins tailored to the cancer cell. Once it successfully infects the T cell will begin sending out instructions in the form of mRNA. The free-floating ribosomes will immediately begin producing the chimeric antigen receptors. Now when we return to the scenario of the cancer cell, the normal antigen receptor does nothing, but our new chimeric antigen receptor is able to identify the cancer cell, and the T cell floods the cancer cell with toxins and kills it. CAR T-cell therapy has minimal negative effects on the patient because it targets specifically the cancer cell. Immunotherapy?